I have a few terms that I need to introduce that belong to Objective 2, and then we'll go on and cover Objective 3. In Objective 2, we looked at the specifics behind carbohydrate metabolism. We saw how one molecule of glucose can be metabolized via glycolysis, the TCA cycle, and the electron transport chain for the production of energy in the form of ATP. These three terms are related, but they didn't fit with what I was discussing, so I kind of broke them off and added them to this presentation. The first term is gluconeogenesis, and my advice is just to look at the word. Gluco, you should think of sugar, carbohydrate. Neo refers to new, and genesis, to make, to produce. Gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors. There are lots of different molecules, different organic substrates that we ingest. We do not live on a diet of glucose alone. So other molecules, such as lactic acid, glycerol, amino acids, various amino acids, can be converted to glucose. And when they're converted to glucose, it's referred to as gluconeogenesis. Glycogenesis, glyco for glycogen, genesis, the formation of. This is the formation of glycogen from glucose. This is a very slow process that does require energy. Glucose molecules become linked together and stored. And this is in the form of glycogen. This is a polysaccharide. And glycogen is commonly stored in the liver. It's stored in skeletal muscle. And it's kind of a carbohydrate reserve. When our blood sugar drops, this molecule is mobilized and it undergoes a reaction that releases glucose into our system. That reaction is referred to as glycogenolysis, or lysis rather, glycogenolysis. Glycogen lysis, the breakdown of glycogen, a very quick reaction that involves a single enzyme. This is our glycogen molecule, or at least a piece of it, and one enzyme will simply split the bond, releasing a glucose molecule. This glucose molecule can then enter the bloodstream, and it can help maintain homeostatic levels of glucose in our bloodstream. In Objective 3, we're going to look at lipid and protein metabolism. I'm not even going to begin to get into any type of detail. I just want you to understand that lipids and proteins can serve as an energy source. When our body utilizes energies, the first energy source, or the first molecule that it metabolizes is a carbohydrate. Carbohydrate metabolism we covered in detail in the last objective. When carbohydrates are depleted, the body starts breaking down lipid. What is the most common, or how is lipid stored in the body? In our fat cells, lipid is stored in the form of a triglyceride. Once the lipid is gone, the body can metabolize protein. But this is something, that the body really strives not to, and that's why lipid storage is so prevalent or so important. Lipid catabolism, we find that there are a number of there are enzymes in the cytosol that convert glycerol to pyruvic acid. And let me just remind you that a I just mentioned that lipid is stored in the body in the form of a triglyceride. And a triglyceride molecule is made of one glycerol and 3 fatty acids. I think I have a diagram of a triglycerol. No, I don't. We've seen it before when we were looking at the organic molecules back in Module 1. But a triglyceride is one glycerol and 3 fatty acids. Glycerol can be converted to pyruvic acid. And pyruvic acid can then enter the mitochondrion, where it can undergo metabolism via the TCA cycle and the electron transport. Fatty acids can also be metabolized or catabolized, and this process is known as beta oxidation. Fatty acids are converted to acetyl-CoA. I didn't talk a lot about acetyl-CoA, but just to try to give you some comprehension of the placement of this molecule and what we've already discussed. At the end of glycolysis, you end up with pyruvic acid. And pyruvic acid, once it enters the mitochondria, is converted to acetyl-CoA. So in beta oxidation, fatty acids are converted to acetyl-CoA. And that acetyl-CoA, again, can enter into the TCA cycle and then the electron transport system for the production of ATP. This is a fatty acid, and if you look at the structure of a fatty acid, it's just a very long chain of carbons and hydrogens. And at the very end, you have some oxygen thrown in. Beta oxidation involves taking this very long chain and basically clipping off 2 carbon molecules that is acetyl-CoA, and that acetyl-CoA can enter into the TCA cycle, and once in the CTA, those coenzymes NAD and FAD can then carry hydrogen atoms to the electron transport system for further breakdown. In terms of protein metabolism, we know that there are upwards of 140,000 different types of proteins, each with a different function, a different structure, and all proteins are built from the same 20 amino acids. They're just in a different primary sequence. When a protein is recycled, the peptide bonds are broken. If you remember, amino acids are linked together via a covalent bond called a peptide bond. And as this protein is being recycled, these bonds are simply broken. And that releases amino acids. And those amino acids can then go back to the nutrient pool, where they can be incorporated in the production of a new protein, or they can be broken down. Some amino acids, but not all, can be broken down and enter the TCA cycle at various stages. It just depends on the specific amino acids. So not all amino acids produce the same result in terms of energy production. It does vary, but amino acids can be broken down. That is the end of Objective 3. Again, I really just want you to understand that proteins and lipids can also serve as an energy source. Many of them can enter into the TCA cycle at various stages. We will cover protein and lipid metabolism in the second semester, but I needed to at least introduce it now.